Okay, so good morning, everyone. So thanks all for coming today to the seminar series in Ecology and Evolution. So as usual, people who attend online, you can report any technical issue in the chat and ask your question in the Q&A tab, and you will be allowed to speak directly in the room to ask your questions. So we just have an announcement regarding the seminar, which is that we have extended the deadline to suggest speakers until the 16th of December. So don't hesitate to send us names and proposals. And today, quite exceptionally, it's my pleasure to introduce Charles Mullon, who will be giving the talk today. So Charles is developing theoretical research that tries to identify the ecological conditions that allow the emergence of phenotypic diversity. So Charles started with a PhD in London, and then this was followed by a postdoc at the Department of Ecology and Evolution in Lausanne under the supervision of Laurent Keller and Laurent Lehmann. And then Charles moved as a lecturer shortly in Japan, and now is back as a group leader, an assistant professor in Lausanne. Thanks a lot for coming, Charles, and I give you the floor for the presentation.
Thank you, Jeanne, and thank you all for being here. So yes, I'm Charles Mullon. I'm come not for very far from Lausanne. And yeah, so as Jeanne mentioned, we have a small group in which we try to better understand the ecological and genetic basis of polymorphism, and we use mostly mathematical and computational methods to do this. And today my talk is entitled Foraging for Locally and Spatially Varying Resources, and this is a project that's recently finished spearheaded by the postdoc Max Schmidt, who is now in fact an assistant professor at the University of Tübingen, as well in collaboration with Klaus Rufler from the University of Uppsala and Laurent Lehmann from the Department of Ecology and Evolution in Lausanne. And the starting point of this research or this project is really quite simple. It's simply the recognition that adaptation to extract resources is a major driver of biological diversity. So here I'm showing really some pictures of animals that show remarkable traits that seem particularly well fitted to forage and extract energy from their environment in order to survive and reproduce. And whilst the traits that are probably most conspicuous as morphological, these traits for resource extraction really span the whole organism from physiology to behavior. And here, obviously, the traits are very obviously different because we're looking at very distant lineages, but these differences can be observed at much finer phylogenetic scale. So diversity can be observed very within species and between closely related ones. So here is an example in three-spine stickleback, which is an emblematic example of such diversity within lineages, or well, within species, where these different this species has diverged between lake and stream habitat across multiple locations in the Northern Hemisphere. And across these spatial locations, these two morphs have diverged into the morphology in order to extract different resources that they encounter. So whilst the lake morph shows additional and longer gill rakers that allow them to feed on zooplankton, the stream morph shows fewer and shorter gill rakers as they tend to feed on benthic microinvertebrates. Another really famous and celebrated example, of course, is in Darwin finches, where here, within the same island, different species of this genus shows highly diverged beak morphologies, with each form specific fitted to a specific resource. Okay, so while you have large beaked finches, which are specialized to hard and large seeds, you have other species that have small pointed beaks, which are more adapted to and specialized to smaller and softer food sources. So in fact, these two examples of the three-spine sticklebacks and Darwin's finches really underlie two main processes that have been invoked to explain resource polymorphism. So where resources vary between habitats, so for instance, when lake and stream offer different diets, diversity here is mostly driven by what I'll refer as spatially disruptive selection, okay? Because different traits are favored in different spatial locations. And so in this case, polymorphism really leads to local adaptation, where each morph shows a better fit to the habitat it lives in. So one way so if you were to characterize schematically the environment in that case, you could, if you had some kind of resource character or property on the x-axis, such as prey size, and the frequency of this resource according to its property on the y-axis, then different patches would show a very peaked distribution around different values. But in contrast, where resources vary within habitats, for instance, where each islands offer a diversity of a wide range of seeds, then diversification in this case is driven by local competition. And this leads to character displacement owing to negative frequency dependent selection, as individuals that feed on less exploited food sources enjoy less intense competition. So one way to conceptualize this is to, again, if we have on the x-axis some kind of resource character or property, on the y, you would have a certain distribution with a certain variance, which is given by the width of the curve. And so on top of this distribution, what you can do is map the efficiency at which a given genotype is going to extract the resources with a given character. So in this particular example, this genotype feeds most efficiently on the most abundant resource, and the width of this curve would give you the niche breadth of this genotype, because it tells of what is the variance of resources it can really efficiently extract. So in this particular example, this genotype can actually efficiently exploit almost all of the available resources, not leaving much space for other traits to coexist. But now, if the niche breadth is actually quite narrow compared to the resource distribution, several genotypes can then coexist within patches, as each will specialize on a given food source. So you have many mathematical models that have looked at these two processes. I've given just a few citations for those here. And one, even though they've made many different interesting points, what I'd just like to underline, what's coming out of these models is really the antagonistic effects of gene flow or dispersal between habitats on these two processes. So because on one hand, when dispersal is limited, this will tend to favor spatially disruptive selection, or favor the emergence of polymorphism under spatially disruptive selection, because it allows different morphs to become associated to the habitats it's most fitted to. On the other hand, when you have locally disruptive selection, so when it's driven by local competition, limited dispersal actually disfavors the emergence of gene flow. And this is because limited dispersal in this case leads to interactions among genetically related lineages, so among kin. And these kin don't show sufficiently diverged traits or genotypes in order to escape local competition. So another way to put it is like kin selection opposes polymorphism. So this is where the literature kind of stands. You have current models that have looked at either of these two processes, but essentially what they've done is that the theory has assumed that resources either exclusively vary within patches or between patches. But probably more realistically, resources vary probably a bit of in both ways. You have variation between habitats as well as within habitats. And so given the antagonistic effects of gene flow on these two processes, the question we had was, well, what happens in a landscape that is heterogeneous between habitats and within habitats. So to answer this, we actually thought of the simplest model we could have that could lead us to some kind of analysis. So we look at a consumer species that inhabits a landscape made up of heterogeneous patches. And these patches are connected to one another via gene flow according to the island model. So this means that if an individual leaves its patch, it's equally likely to land in any other patch. Okay, so there's no isolation by distance in this model. All patches are assumed to have the same carrying capacity so that we can really focus on the heterogeneity on the resources. All right, so there's no effect of having sinks or sources of individuals. The population follows a simple seasonal life cycle whereby individuals first exploit the resources locally. Then, according to the amount of resources that they've accumulated, then produce offspring. So individuals that have accumulated more resources will produce more offspring. Offspring then either disperse or remain in their natal patch. And adults in the meanwhile either survive or die to the next year. And if they die, they open breeding spots in their patches, which are then competed for by the offspring that are present. All right, so just a bit more to explain a little bit more about the model for the terms of the resource. So we assume again that it varies in some kind of relevant character or trait. So you could think of prey size, or thinking, I like to think of the hummingbird for this example, where the resource property could be the length of the corolla tube or the flowers that carry nectar. And here, for instance, I have shown two distributions for two types of patches, where patches of type 1 have on average shallower tubes, and patches of type 2 have on average larger tubes, deeper tubes. And this difference among averages gives us then some kind of variance among patches. And since both types also show variation within patches, you have some measure of variation within patches. And the sum of these two sources of that variation gives you the total variance in the resource property across the landscape. So for the purpose of this talk, I'm showing a simple landscape made up of two distributions that can normally distributed, but our results are more general and apply to any kind of distributions. In any case, once you have decomposed the variance in resources, you can quantify the amount of heterogeneity and resources in the landscape through what we've labeled as EST, which is simply the variance between patches and resources divided by the total variance. So you can see it as kind of the ecological differentiation or patch heterogeneity in links to QST for phenotypic differentiation or FST for genetic differentiation. So when EST is 0, as shown as the bottom graph here, this would mean that all patches have the same distribution of resources. When EST equals to 1, we recover the case where each patch does not show any variance within patches, and all the variations consumed between. And when EST is intermediate, this means you have variation in both. So this allows you to tune from the two extreme models that are currently in the literature and to investigate what's in between. So what about consumers? Well, consumers, we also assume that they vary in a relevant quantitative character that modulates the efficiency with which they can extract a resource with a given property. Okay, so in other words, some trait values are just simply better at extracting certain types of resources. So if you go back to the hummingbird example, a hummingbird with a bill length of 3 centimeters will be most efficient at extracting nectar from flowers with a tube of 3 centimeters, just very simply. So we assume that this relationship between the consumer trait and the feeding rate is given by some kind of normal distribution, where the mean of this distribution gives a character that is expressed by the individual. And this normal curve has a variance, and this variance then gives you the niche breadth. Okay, and we assume that this is fixed for all individuals. So the greater variance means a wider curve, and therefore a greater capacity to extract a wider range of resources. But this is fixed and does not evolve. Okay, so then, given their feeding efficiencies, individuals compete within patches for resources. And to model this, we assume that during a season, resources are consumed over a continuous time range, during which individuals extract and deplete resources at a rate that is given by their trait and the trait or the character of the resource it is attacking. So this gives you a kind of a typical lottery type of competition that you can model using differential equation over time. So here I've shown the rate of change of a certain type of a certain resource J, which is depleted according to the efficiency of all the individuals in the patch. In the meanwhile, the energy accumulated by each individual I, so EI, is simply the sum of all the resources it is extracting from the environment at a certain rate. So in this model, what you could imagine is that the resource is kind of a liquid with a given amount, and each individual has a straw, and we all put our straw in this liquid and start sucking on it, and the one with the highest suction will be able to have more resources than others. So you can solve these differential equation to give you the total amount of resources extracted by each individual over the exploitation season. And so, for instance, here I've shown an example where the individual 1 has a greater efficiency at extracting the resource than individual 2, so that whilst the resource level drops to 0 over time, the energy of individual 1 accumulated by individual 1 through that resource is greater than individual 2. So of course, the total amount of resources that you can get depends on the amount of time within the season that you have available to extract resources. And for now, I'm going to assume that this exploitation time is long, so long enough for the resource in each patch to be depleted. And I'm going to further assume that this total amount of resource is the same in each patch. Okay, so even though patches can vary in the type of resources that they carry, the total amount is fixed. So what that means for now is that each patch is going to produce the same number of offspring because all the resources are depleted. So in other words, for those who know the term, this selection is soft for now. We have soft selection. In terms of genetics, I follow really typical adaptive dynamics or quantitative genetics assumption in the sense that I assume that it's genetically determined by a quantitative trait locus. And this locus evolves via rare mutations with weak phenotypic effects. And for now, let's assume that individuals are asexual and haploids, leaving the complications of sexual reproduction for later. So under these assumptions, we know that trait evolution kind of occurs in 2 time steps. Okay, and first, the population evolves gradually under directional selection via the constant input of mutation and the action of natural selection. And the population in this case will eventually converge to some kind of equilibrium. Once the population expresses this equilibrium, you can either, 2 things can happen. Okay? You can either experience stabilizing selection so that the population will remain monomorphic for this equilibrium, or the population will experience disruptive selection or negative, more precisely negative frequency dependent selection to become polymorphic so that the trait distribution will go from unimodal to bimodal. And this is kind of the outcome that we're looking for, because in this case, this means the maintenance of polymorphism. So over to results. What we find is that first, the population converges to express the trait value that matches the global resource property. And this falls in line with previous model of soft selection, actually. So at this point, initially, all individuals express the same phenotype, and the population is not better adapted to any specific patch type. Once the population expresses this equilibrium, we find that whether diversity or not depend on whether this relatively simple condition, which is why I thought I'd show it, is fulfilled. So if this is fulfilled, you have condition for polymorphism. And I'm going to run through it. I just want to remind that EST measures the level of patch heterogeneity in resources. So if for now we ignore what's within square brackets and just look at the what's on the outer left and the outer right, what we see is that polymorphism is emerges most easily when the total resource variance is large relative to the niche breadth. Okay, so this is really intuitive and in line with all previous models. It simply reflects that the greater the resource diversity, this provides more ecological opportunities and facilitates the coexistence of specialized consumers that have a narrow niche breadth. All right, so looking within the square bracket now, we see that the variance that's on the outer left can be decomposed according to its between and within patch components. And each of these are weighted by a quantity chi, which basically that's squiggly x. So these two quantities, chi1 and chi2, are relatively complicated functions, but basically they only depend on the demographic parameters. So they depend on patch size, adult survival, and dispersal. But what they vary most with is with dispersal. So I've shown here on the graph. So the first term, which is proportional to patch heterogeneity EST, captures how polymorphism can be driven by spatially disruptive selection, okay, favoring local adaptation. And on the top right here is shown the function chi1 that weighs this. And you can see that when EST is large, dispersal will therefore tend to inhibit polymorphism. And this reflects the well-known notion that gene flow inhibits local adaptation as it homogenizes genetic variation between patches. Note also that chi1 tends to decrease with patch size. And this is because when you have smaller patches and dispersal is limited, you have greater kin competition. So this means that simply that an individual's reproductive success comes at the expense of kin. So generally, selection on competitive traits tend to be lower in this case. And so this is why you see this effect. And even though I haven't shown it, adult survival has the same effect. All right, so if we now look at the second term within square brackets, this captures locally disruptive selection, or more specifically, negative frequency dependent selection within patches at the local scale. And this selection favors rare morphs in each patch because these morphs are able to exploit resources that are under less intense competition. We can see that the strengths of this selection compared to now increases with dispersal. And this is, again, as shown in previous models, because when dispersal is limited, the individuals interacting with one another tend to be genetically related, therefore expressing the same traits, and therefore they cannot benefit from exploiting different competition. They cannot escape local competition. So this equation is nice because it's simple, and also because it really allows to connect classical models of local adaptation with classical models of local competition. Okay, if you tune EST from 0 to 1, you obtain all the other models, and we've shown that you recover all the existing equations. Now, seen as both types of variation have additive effects on the condition for polymorphism, what we can take out of it also at a broad scale is that spatial and local variation both contribute together to diversity. This may seem intuitive, but this is not necessarily what we had expected. Although, even though that is the case, it doesn't mean that both types of selection sit on an equal footing. So to see this, we can look at the parameter space that leads to either polymorphism, shown in blue, and this is what a typical simulation would look like over time, where each individual is a dot, or monomorphic, where or the case where the population state remains monomorphic, as shown in the bottom. Okay, so here you have dispersal on the x-axis, patch size on the y-axis, and this is the case where EST equals 0, so all variation is within patches. And as expected, we see that polymorphism is favored by high gene flow. But as we increase patch heterogeneity and in here I'm keeping every other parameter constant, I'm only changing EST. This parameter region for polymorphism changes so that when EST is 1, and now all variation is between patches, now polymorphism is inhibited by high gene flow. So what this sort of reinforces the idea that whether or not you get polymorphism depends on the level, the distribution of variation of resources and its interaction with dispersal and patch size. But I'd like to highlight that actually dispersal tends to inhibit high gene flow even for very low level of patch differentiation. As you can see here from EST 0.01, you already have inhibiting effects of high gene flow. So what this means really is that spatially disruptive selection tends to be stronger than locally disruptive selection. So I'll just to sort of wrap up these few results. We saw that polymorphism in a consumer traits emerges more readily when resources vary both within and between patches. In other words, when selection is both spatially and locally disruptive. But kin selection, due to limited dispersal, by contrast, will oppose polymorphism, especially when polymorphism is driven by local competition. And this is, again, because local interactions among kin reduces the advantage of expressing alternative phenotype to escape competition. So although both spatially and locally disruptive selection contribute jointly to polymorphism, spatially disruptive selection is typically stronger than locally disruptive because it's less hindered by kin competition. So one broad conclusion at this stage that we could make is that polymorphism in a patchy landscape would result more easily from local adaptation than from local competition, although both contribute to the evolution of phenotypic variation. Now, both types of selection may favor polymorphism, but they won't lead to the same distribution of phenotypic variation across the ecological landscape, of course. So to see this, we can consider the phenotypic variation in the population at the mutation selection equilibrium in some simulations. So I just want to show on the top here, you have the distribution of unique phenotypes or genotypes for different levels of dispersal at mutation selection equilibrium when EST equals 0 on the left column, EST equals 0.5 on the right column. And the blue region is where the analysis predicts polymorphism. And in the pink region, or here rather orange region, you monomorphism. So just at a quick glance, you see that the simulations agree quite well with the analytical predictions. But what I want to rather look at is the bottom graph, where is mapped the variance in phenotype in total in black, where you see that when resources vary only within patches, so when EST equals 0, the variance in the population increases in the trait increases with dispersal. But when you have some patch heterogeneity, actually it decreases with dispersal. Now this total variance you can decompose to obtain what is referred to as QST, which is the between patch genetic variance divided by the total genetic variance. And that gives you the kind of an idea of how much phenotypic differentiation there is in the consumer species across a landscape. And what's particularly interesting, and it has been suggested and done many times, that you can compare QST, which is presumably under selection, with FST, which is at neutral markers, so genetic differentiation at genetic markers, to kind of try to understand the operation of selection on phenotypic traits. So we computed FST from our simulations, where in addition to the QTL, there was well, here it doesn't matter, but unlinked genetic locus that evolved kind of like a microsatellite marker, at which we computed neutral differentiation. So here is shown QST in black and FST in gray, but this is not really what I want to focus on. What I'd like to focus on is rather the dots, which represent the difference between QST minus FST. I realize that this is quite small. I don't know if you can see it from the room, but focus on the dots, and the dashed line is giving you where QST equals FST. Okay, so if we focus on these circles and where they sit compared to this dashed line, we can see that these comparisons between QST and FST fall into like three categories. So in the peak region, where selection is essentially stabilizing for the same trait value across the landscape, QST is sort of equal as FST. And here I'm only showing for a few simulations, but we have more results to show this. In the blue region on the right, where you have some level of patch heterogeneity, QST is greater than FST. And this QST is greater than FST. Well, in contrast, when you have polymorphism and there is patch homogeneous, so polymorphism is due to local competition, QST is lower than FST. Okay, so our observation that phenotypic differentiation exceeds neutral genetic differentiation under local adaptation falls completely in line with previous theory and suggestion. But another common idea is that QST lower than FST is an indicator of stabilizing selection, okay, when selection favors the same trait value in all patches. So actually what we find is that phenotypic differentiation in this case is actually similar to FST. So phenotypic differentiation is similar to neutral genetic differentiation when the trait is under, I guess, weak stabilizing selection. Rather, where we find a strong signal of QST being less than FST, this is when selection is disruptive due to local competition. And this is because negative frequency-dependent selection at the local scale generates greater phenotypic variance within patches compared to neutral genetic variance. And this is actually an idea that was proposed by Jean-Baptiste Lamy earlier, but it was just written in a review, but not really followed up that I know of. But and this is interesting because some simulations by Michael Whitlock and Frédéric Guillaume actually found that when you have this spatially uniform selection, it tends to lead to weak signals of QST less than FST. And the only time you get a strong signal is when you have really big mutational effects, really big mutations that have large phenotypic effects. So in essence, in most cases, the statistical methods have a relatively low detection power. But signals of QST less than FST have been reported in a few natural populations and for a relatively wide range of traits. It's not common, but it happens. So now an alternative suggestion that building on Jean-Baptiste Lamy is actually that these traits perhaps are under negative frequency dependent selection, or what population geneticists would call balancing selection. And again, this is because such type of selection will lead to greater variation within patches compared to neutral variation. So if you recall, I assumed that individuals had enough time to forage all the resources within patches. Okay, so that means that all resources were consumed within the patch. So each patch produced the same amount of offspring, so that selection was soft. So we can then ask quickly what happens when in fact exploitation time is shorter, so that patches in which individuals are better adapted to the local conditions can extract more resources, and therefore produce more offspring. Okay, and this leads to hard selection. So in that case, we find 4 possible outcomes for evolutionary dynamics, which are represented in the 4 different types of color of this quadrant, where you have dispersal on the x-axis and patch differentiation on the y-axis. And just briefly, when dispersal is high and patch heterogeneity is low, we see that the population, as before, remains monomorphic to the average type of resource. So here you don't have polymorphism, but the population adapts to the mean resource in the global landscape. By contrast, if we go to the opposite, where patches are very heterogeneous, the population also remains monomorphic, but in this case, they will end up being adapted to a single type of patch, and this depends on initial condition. Okay, so if the population is initially more adapted to patch type 1, like I've shown here, it will gradually evolve to be simply adapted to patch of types 1. And this is due to the fact that patches where individuals are better locally adapted produce more offspring. Okay, so as a result, if individuals are initially fitter in one particular type of habitat, they send out and swamp the population with a certain type of offspring that is adapted to one patch type, leading the population to be adapted to a single patch. So in essence, the population is kind of stuck there, adapted to a single type of habitat, because the fitness landscape becomes too deep to cross. But if we move down from this scenario, we see that when patch differentiation is intermediate, so when there's a little bit of variation of resources within patches, the population experiences disruptive selection and thus becomes polymorphic. And this evolution to polymorphism can happen either via a transitionary period where you become more adapted to a patch type, as shown in the lighter blue, or as before, if you converge to the mean. But if we kind of ignore these specific dynamics, what I find interesting here is that having just a bit of ecological variation within patches allows the population to escape the evolutionary dead end of being adapted to a single patch. And you don't necessarily really need a lot of variation within patches for this to occur. And this has potential implication for ecological stuff like migrational meltdown, which may be easier to escape than previously thought. So the analysis I've shown really only answers whether you get monomorphism or polymorphism, and I've only shown example where you had 2 types, all right? But of course, you can get cases where you get many more morphs that emerge and can be maintained, as shown in this particular example, where you have 11 morphs eventually coexisting in the population. And you could get multiple morphs when there are multiple patch types. So you have the population adapting to different 11, in this example, 11 different types of patches, or when the niche breadth is much, much narrower than the variance in resources, allowing for many specialized consumers to coexist in similar patches. And in principle, again, especially disruptive selection would favor as many morphs as there are patch types, and this will be facilitated by limited dispersal, whilst locally disruptive selection favors as many types to fill the local niches, and this number should be inhibited by limited dispersal. And this is really what we find. Here you have dispersal on the x-axis and the number of morphs in the end, under cases where you have much more variance in the resources. And you can see that this fits this prediction. Okay, so on a last note, just a quick note on sexual reproduction, because our model assumed clonal reproduction, but previous theory in any case suggests that the condition for polymorphism, the mathematical condition, actually predicts also very well whether or not you get polymorphism in a sexual diploid species where the trait is has an additive genetic basis. And this is this is what we found in individual-based simulations, and except that in this case, as you can see, that in a situation where you only have 2 morphs, so you have like 2 peaks in your trait distribution, now we have 3. And this is really just because you have these divergent genotypes that create, in this case, hybrids. Even though they are more numerous, they're actually less fit because they cannot extract as many resources as the other 2. So because these hybrids are less fit, selection should in turn favor mechanisms such as allelic dominance or associative mating to avoid the creation or the formation of these intermediate phenotypes. So in fact, the project, this is where we're taking the project next, where we are investigating the evolution of genetic and behavioral mechanisms that can avoid the production of these unfit hybrids. And I think it's especially interesting because the evolution of these different mechanisms, at least intuitively to me, will depend on dispersal and gene flow, but also on whether selection is more or less spatially or locally disruptive. And this would have potential implication for speciation and diversity. Okay, so takeaways from this talk are thankfully few. The first is that I hope I've conveyed that polymorphism in a consumer trait emerges more readily where resources vary both within and between habitats. And secondly, perhaps although this theory is fairly abstract, and I would be very cautious in trying to make specific empirical statements, I think it leads to at least one perhaps interesting quantitative insight, which is that it may be useful to consider that when phenotypic differentiation is significantly lower than genetic differentiation, this may be indicative of negative frequency dependent selection at the local scale. Of course, this is probably most relevant for traits that mediate competition. So I wouldn't assume that this is the case for every kind of trait. But more broadly, really, I think this research just reinforces the general notion that a competition for resources is a major driver of adaptive variation and thus participate to the diversity of life forms that we observe. So on this, I'll thank my collaborators again, the Department of Ecology and Evolution at the University of Lausanne for hosting me, and the Fondation Suisse for funding and for your attention. Thank you. Yes.
Thank you very much. I was wondering, the population genetics you use for deriving the polymorphism criterion is adaptive dynamics, where you have a very narrow population is narrow around some resident trait character value, and then you shift it by an infinitesimal change and like iterating games, or is it, what is the method for?
Yeah, exactly. It's an invasion analysis.
Okay. So then I didn't get how you then plug back within population and between population genetic variance.
So this was through individual-based simulations.
Okay, but the criterion for polymorphism invasion, there was no genetic sigma in your criterion for invasion, basically.
No.
It does not depend on it, or at least as long as it's narrow enough, you don't see any.
No, exactly.
Okay. And did you explore the simulation to go for wider, more polymorphic within population situations where adaptive dynamics may be still qualitatively okay, but quantitatively?
So you mean larger?
Well, you will have a genetic variance that is quite substantial within population, within habitats, within populations.
Yeah, so we did, and so what you find is essentially maybe the polymorphism or the distribution is not as double peaked as pretty with these two clear peaks, but they tend to be they tend to get closer to one another, and you don't have such a shallow trough between the two peaks. But you still get two modes, yes, except when you have sexual reproduction, then messes everything up. So in that case, if you have sexual reproduction, recombination, multi-loci, then you just get a blob. You don't observe. But the genetic variance is still a bit higher. So if you look at the phenotypic variance, how it changes in sexual diploids with recombination, multi-loci, a large number of additive effects, then you still see that when the condition for polymorphism is fulfilled or not, there is a bump, because really selection still drives a bit more variation than you'd expect, but it's not as clear.
Yes, I was intrigued about this result about the contrast between QST and FST, and I was wondering whether this result also depends on asexual reproduction or not, because I kind of remember finding cases where you could have this QST below FST, even with disruptive selection in different patches, even if you had different optima in different patches, because it depended on the strength of drift compared to the strength of divergent selection. So I remember of previous studies concluding just the reverse, that actually if you find that, you can't really say that you don't have disruptive selection between patches,
really?
Yeah.
So yeah, I'm not aware of this.
Okay.
Here we've used simulations with a big population, so I guess the effect of drift is probably small. So it's 20,000 individuals in the population. In sexuals, we still
drift in the patch.
Drift within the patch.
Yeah.
Okay. So here drift is very high in the patch, because we have and I can show maybe the. So at least for the all the analysis that we did still showed this pattern, that when polymorphism was only driven by local competition, and you had polymorphism in sexuals as well, you observe QST that less than FST.
Yeah, what I mean is that if you have
you could have this.
Yeah, in this situation where you have some heterogeneity between patches, if depending on the relative size of this divergent selection and the strength of drift, you could have sometime neutral divergence higher than the variation you would expect maintained by selection.
Okay, that's interesting. We haven't observed it in the parameter space that we explored.
Okay.
But maybe
yeah, it would be interesting to test. And also I wanted to know if the Chi1 and Chi2 parameters you have, whether they have any interpretation in terms of population genetic parameters like relatedness or FST or something like that. Can you relate them to classic measure of neutral differentiation in a way or not?
So you can, you can. So there are actually, in terms of pairwise and three-way relatedness, so you can actually decompose, but they're very hard to read, because I think they combine both, select kin competition as well as selection, and you don't see it essentially from just looking at the expression in terms of relatedness.
Okay.
Maybe we could decompose it like more clearly, but we haven't managed. Okay. But we have an analytical expression only in terms of relatedness parameters. Yeah.
Thank you.
Thank you.
Okay. You hear me? Okay. It's a fake microphone. So thank you for the talk. So I wanted also to talk about the Chi1 and Chi2 parameters. Basically, because I was not quite clear about the role of kin competition in the model, because a lot of the previous theory has actually also looked at patches with basically well-mixed populations without kin selection operating in each patch, coupled with migration, and has looked at the effect of migration on the emergence of monomorphism, polymorphism, and so on. So I was wondering if you set like relatedness to 0 in your Chi1 and Chi2 parameters, do you still get the same kind of qualitative conclusion, or is it really dependent on kin competition?
Yeah, so actually with here I didn't show it, but this, so this general equation, here, so as it depends on patch size, adult survival and dispersal. And if you put in values that have been, that fit different models that are existing, you recover exactly the same equations or the same mechanism proposed. So in particular, Chi1, for instance, if you say, okay, I want to recover the models of local adaptation, I put EST equals 1, so only Chi1 matters. And most of these, actually these models often consider just 2 patches that are of infinite size. So there's no kin competition. And in fact, here we recover the condition that this is 1 minus 2 divided by m, which is exactly what these other models have found. I don't know if this was exactly the question, but.
The effect of migration what itself
yeah. Ah, yeah. So on Chi1 then. So kin competition can be seen from the graphs where you see the effect of patch size here on Chi1. So you see that here it's on a log scale, huh? So if you go from a patch of a size of 100 to 10, kin competition really reduces the strength of the effective strength of spatially disruptive selection.
Yeah, but the relationship with Chi1 and migration, for instance, it is the same for Chi1 and Chi2.
Yeah.
Irrespective of the order and the.
Yeah, absolutely. So here you have kin competition effects that act differently. So because here, with respect to dispersal, the effect on Chi1 is not due to kin competition. Actually, it's really for the different genotypes to be associated to different patches. So selection really benefits from dispersal being low for polymorphism to emerge. However, you still have kin competition effects within patches, and this is captured by the effect of patch size. In contrast, here everything is due to kin competition. Okay, so you have for Chi1 there are two effects, but yeah.
Thank you.
The mic. Right?
Yeah, the infinite is close to, it's actually quite close to the n equals 100, but yeah.
You can see so if you would add the infinite population size on this graph, you would see that it would be flat on the for the first one, but not flat for the second. For the first, not flat for the first one, but flat for the second. So it could be a nice way to
okay, yeah, that's a good suggestion. Thank you.
Question in the chat? No? In the room? Yeah.
Okay. When you discuss sexual populations, these are sexual diploid populations. So you can have two effects. In fact, the effect of diploidy and the effect of recombination on a multi-locus basis. Can you say different things about the distinct effect of these two factors?
Right now, no, because most of the simulations we did, it was a single quantitative trait locus, so recombination played zero role. But this is something we're exploring next, actually. So I hope to have more things to say next time.
I have a question. You use the island model.
Mm-hmm.
What do you think can make the spatial structure in the migration?
So I guess you would have different effects of having explicit isolation by distance, and it would depends on how strong the transition from different patches is. In a way, here I think I've made the condition probably the most difficult for local adaptation to emerge, because you can really have this migrant pool that falls everywhere. So the effective dispersal would be very large if you had isolation by distance, but different pockets. So intuitively, I think that qualitatively, the results wouldn't change. They would have no effect to have isolation by distance, except that maybe making the conditions for local adaptation a bit easier, and for negative frequency-dependent selection a bit harder at the local scale.
maybe it can change when you add recombination, because if you use different routes at different place in space, then you can generate by recombination larger possibilities of trait values. No? What do you think?
I think that is true. Do you think it would have an effect on the eventual point at mutation selection equilibrium, or rather on the path taken for adaptation to emerge? So do you think this is, to me, it's not clear, at least, if whether this is an effect on the transition dynamics, or it would have an effect on the equilibrium distribution?
You know better than me, but I would say also on the equilibrium.
Also on the equilibrium.
But you can have a contrast between the local effect and the global effect, and that you don't have here, because
absolutely. Thank you.
Yeah, hopefully.
I was just thinking that the fact that the local differentiation, the relationship between QST and FST can be reversed. This is also something that is discussed for cases where you have a strong balancing selection within patches, like for self-incompatibility alleles, for instance. So anytime you have strong, frequency depend.
So you said this is.
So for instance, it was described for alleles of self-incompatibility that the level of differentiation between population can be less than you expect by random drift, because you have the same mechanism as in your model, a force that is maintaining a local polymorphism more than drift would.
No, that's interesting. Actually, there was also here the ecological scenario is one of resource competition. So I've only discussed this, but there are many different other processes that could, alongside local adaptation, can actually increase effective migration rate through negative frequency dependent selection at the local scale. So, for instance, if you had local pathogens adapting to the most abundant genotype which you find in a patch, this creates negative frequency dependence locally, but you can still, but if you have local adaptation acting on top, this actually inhibits local adaptation at a local scale. I think this was described by Daniel Bolnick in sticklebacks, actually, but incompatibility also. Okay, but you say they measured and they made the statement that QST lower than FST. Okay.
Sorry, I think there were papers. I think it's Sylvain Glemin, I think.
Okay.
I had a paper about that. But
he was in Lausanne last week, actually.
Yeah, I'm not completely sure. But
I'll ask him. Thank you.
Another question?
Hello. So I was wondering, because most of the interesting part of the model is really when you balance this kin selection against local adaptation type of effects. And it seems to me that much of the effect of kin selection is really, at least in part, caused by how much is it costly to be having a slightly different phenotype, considering the resource that you have locally. And in your particular model, something is probably doing a lot of effect on this is really the shape of the fitness function. See where I'm going, and so because it's really flat in your case, probably you could imagine that it would be very different if this fitness function relating resource to fitness was not as flat as in the Gaussian that you use. So perhaps completely changing the balance between kin selection and the other local adaptation process here. What do you think?
Yes. But in that case, it would make actually kin selection even. I mean, the deleterious effects of being similar would be even greater, no?
Yeah, probably. Yeah.
Yeah. So
Yeah.
even making the effect of kin competition.
Yes,
even
probably.
stronger.
Yeah.
Yeah.
So because as far as I understand, in your model, you always emphasize the case where EST is very close to 0, where something is really a bit different.
I think that's because I'm more interested in this case. But
Yes. So perhaps it's a very narrow region, but without those assumptions, it would be different.
Maybe you could widen this region. Yeah. Thank you. That's good.
Thank you. Do we have questions in the chat now? No? So thank you very much again for
Thank you, everyone.
And don't forget to propose